So we're going to discuss electrophoresis today. Let's see here. Participants can see you now. Thank you. All right. So this will be a kind of a mixed source because I've taken some things from the old chapter 6, but primarily the flow comes from the book and it's section 5.4. We'll actually start talking about other stuff in chapter 5 next week. And then I'm going to briefly discuss this 69 Weber and Osborne paper, the 70 Lemley paper, and the 75 2D gel of Ferrell paper. I gave you some other stuff to read just so that you would have a sense of history. If you haven't read what I posted, the stuff on the history of gels, you might read that. In my world, that's all current stuff because I was already in graduate school when a lot of that stuff was happening. I may add a question to the to the assignment list if I remember to do it. Let me write a note to myself now. Okay, so the beginning of chapter 6, I guess, that version goes a little bit further with the derivation at the very beginning. And so what it does is it starts with the fact that biological polymers tend to be charged. Certainly proteins and nucleic acids tend to be charged. Okay. And so this is the mechanism by which electrophoresis works. If you put a molecule in an electric field, it will migrate depending upon the direction of the electrodes. And it will move in some environment. Okay, and I'll go through some of this during the presentation, but initially this was free boundary electrophoresis. There was literally no matrix. There were no such things as gels, polyacrylamide gels or agarose gels. And so the original work in the sort of 40s was done in a free boundary apparatus. The other thing that can happen is that the molecule can become oriented if the charge has some kind of asymmetry on the surface of the molecule. And so measuring that orientation is also something that various techniques allow you to do. I won't say very much about that at all. So this is a general way to think about fields and an applied field and how macromolecules respond to an applied field. In the presence of an electric field, there's a force on the molecule, and the force depends upon the charge, Z, so the number of electron units of charge. It depends upon the magnitude of E, which is just the magnitude of an electron in electron units, basically. And then E, big E, is the field in electrostatic units of potential per centimeter, so the magnitude of the field. And then once there's a force on the molecule, okay, it responds by moving. And so there's a velocity that occurs. And as soon as there's motion, there's resistance to motion. So there's a frictional factor, which we call a frictional coefficient. So the velocity then comes out of the force being equal to frictional coefficient times V for velocity. And then we rearrange this equation, and this is something we'll do the next time we talk about this with centrifuge forces, so that it's velocity per unit field. So you take E to the left, and you get V over big E, and F to the right, and you get ZE over F, okay? And then you define this velocity per unit field as something called a mobility, okay? And in this book, it's called a U, or in these chapters, it's called a big U. So velocity per unit field is called a mobility. And the mobility depends on the net charge divided by the frictional coefficient, the factor that resists motion. Okay. If the particle is a sphere, then by definition, the frictional coefficient is 6 pi eta r, where eta is the viscosity and r is the particle radius. We'll call it the hydrated radius later, or the Stokes radius later. Okay. And then that's basically as far as you can go with a simple sort of way of thinking about things. Macromolecules are seldom spheres. Some of them are. A lot of viruses are actually spherical. Coronavirus is actually a bumpy sphere. But generally speaking, you end up having to correct this for shape and hydration. So that's why I say this will be defined as RH or RS. And then it may not be a sphere. It may be some other kind of structure. And then we may end up using an R that is equivalent to a sphere. So if this particle were a sphere, what would its radius be? And then on top of all of that, you have to be aware of the fact in electrophoresis that you necessarily have ions, and the ions necessarily associate with the charge. And so you have an ionic atmosphere, and that immediately creates a change in the net charge because these charges are screened by the ionic atmosphere in the solution and around the macromolecule. So that's sort of a very simple mathematical way of thinking about this. So I think in the book, this is a little bit clearer. I don't have a good Xerox copy of this. In free electrophoresis, you set up your electrodes with a positive and a negative. And I'll say something about this later, but how you set this up, which electrode is at the left and the right, or the top and the bottom, depends on what it is that you're electrophoresing. And so if it's a positively charged molecule, it will migrate towards the cathode. If it's a negatively charged, it will go towards the anode. And you have to set it up so that things migrate in the way you want them to migrate. When we do SDS, polyacrylamide gels, the SDS brings a negative charge, and so everything will go towards the left, and so that has to be accounted for. In free electrophoresis, you basically have to assume that all of the molecules are going to move in a certain direction, and some of them may move to the left and some may move to the right, and you may have two sets of boundaries. The bottom is meant to represent the fact that the solution has other molecules associated with it. And in a better picture of this, the positively charged macromolecule has negative charges around it that are clustered close to it, and therefore screening the charge or the effective charge on the molecule, because these negative anions are changing the net charge and changing the mobility. So electrophoresis can be very sensitive to the concentration of the salt that you include in the solution. And you have to be careful not to have too much salt. Otherwise, the only thing that migrates is the salt, and the macromolecule actually doesn't migrate hardly at all. Okay, so this is a simple sort of design, and it shows in a pictorial way what is sort of going on. Notice the mobility to the right is counterbalanced by some kind of a frictional force, and that's the minus FV. So that's the frictional force, the displacement of ions going in the opposite direction as you move cations to the right. So negative ions have to sort of move the other way for balance. And so there's a frictional force and a electrophoresis force acting on the molecule. And that was that equation up above where you set these equal to one another at steady state. Okay. Now, correcting for hydration and shape can be done partially by applying what is called the Bihuckle. And I've mentioned this to you before, and we've gone over some of this a little bit. We're not going into a lot of detail. But what you can see here is that the mobility can be represented as the ZE divided by the frictional coefficient. If it's a sphere, this is the shape that we use, 6 pi eta r. But then there's this correction factor that comes out of the Bihuckle. The correction factor involves something at the top called a Henry's function. They plot it in chapter 6 to show you what it looks like on a log scale. So you can see it's a factor that only goes from 1 to 1.5 on a log scale. And then the denominator involves this 1 plus kappa r, and kappa has this definition down below. It's known as the reciprocal ion atmosphere. So it depends upon the salt. It depends upon the ionic strength. So the concentration of the salt, square root of the ionic strength. It depends upon these constants, 8 pi, n is Avogadro's number, E squared divided by 1000 kT. But also down here is the dielectric constant. So it depends on the dielectric constant. So you get dramatically different answers if you're in water as opposed to if you're in some kind of a weird solvent, which is often the case. Not in biology, but in other kinds of systems. Okay, and so this kappa generally just what you need to know, it depends on the square root of the ionic strength. Okay, and this reciprocal ion atmosphere, an equivalent word for it is Debye length. So it's the Bihuckle theory. So the Debye length basically says how far out into the solution. Sorry, I'm going to move something off of my tablet. How far out into the solution. So if you have some kind of a charge, how far out into the solution do you sense the charge in this molecule? And if this is a positive charge for the example up above, and we have negative ions out here that depend upon this ionic strength square root, then the Debye length gets shorter in a way that depends upon that ionic atmosphere. And so you don't sense the charge. You can't feel it at great distances because there's a salt atmosphere, an ion atmosphere that interferes with you sensing this net charge. Okay, and so that's what we're talking about. And the Debye length is extremely long at low ionic strength in the absence of salt and very short in the presence of salt. Later, maybe, I'm still not sure about timing of things. If you have solutions that are very dense, like cellular environments, then Debye lengths become critical because everybody is really, really close. And this distance means that they sense charge even in the presence of high ionic strengths. So if these distances become nanometers or less than nanometers between particles, then there can be large charge-charge interactions that are going on, regardless of the Debye length, simply because they're close. They're essentially touching one another. In dilute solutions, the next molecule may be out here. They don't really see one another. And so this Debye length becomes less critical in some ways, although this is the reason why salt can suppress interactions, because they basically don't see one another. Under these conditions, they're going to see one another, and there's almost nothing you can do about it. And then the question is, why don't they clump, and why don't they interact more than they do? So that's a little bit of an aside, but that's what we're really talking about here in a general sort of sense. Okay, now the free solution examples, or the example, was developed by a guy by the name of Tiselius. A lot of the early history of things in electrophoretic and sedimentation transport were done in Sweden, and Tiselius was one of those guys. And this is a Tiselius electrophoresis apparatus. So this is a free boundary instrument, and it's essentially a U-tube. Okay, and the U-tube allows you to have two menisci, basically, where you have an interface between the protein solution, or whatever the macromolecule is, and a buffer. Okay, and you have this ability to create sharp interfaces, and so it's accomplished by having these plates that are allowed to slide so that you can create sharp boundaries. I'm not describing it very well because there's not much detail in the book. This apparatus probably doesn't exist anywhere in the world anymore. There's probably examples of it in labs in Uppsala in Sweden. I don't think I've actually ever seen one of these myself. And so you get these boundaries, and then you turn on the electrophoretic field. It's not even specifying here what direction is what. It's not telling you that. But if, for example, we did exactly what that other figure did, and we turned on negative charge on this side and positive charge on this side, so it'd have to do with these electrodes, then you would start getting migration. But the migration would be the positive ions going this way, and then you would necessarily have negative ions going this way, or macromolecules going this way, but it would also apply to the free salt. And so you would end up having two boundaries. And the way it's often described is ascending and descending, because you have stuff that's going up one side of the tube, but it's also going down at the same time. And so you're sort of monitoring what's happening. The monitoring is usually done with some kind of an optical system that looks at gradients. And so you usually get something that's reflected in peaks. And so you have ascending and descending boundaries. This is a hypothetical mixture. Notice that it's a mixture of the same stuff, but for reasons that are discussed, now I'm probably not going to go into all of this, but basically you get boundaries that move up and boundaries that move down. And a certain amount of this has to do with the fact that macromolecules are an ensemble of charges. And so different molecules might move in different directions simply because it's a subset, or there's heterogeneity in the sample. Okay, but you can see moving boundaries. A lot of this early work was done on readily accessible proteins. So we're talking about the 40s. And a lot of these were blood proteins. Okay, so things like hemoglobin, things like antibodies, IgGs, and they would move in these sort of single boundary directions, and then there would be some extent of resolution. But notice that there would be a lot of broadening, and the broadening would diminish the amount of resolution. And that's something that we're going to talk about as we move through this today. So this was sort of the earliest examples of electrophoresis. High concentrations and the awareness that macromolecules had charge. Appreciate this is pre-DNA sequencing. This is pre-protein sequencing. There's a lot we didn't know at the time. And so to just see that there was something that seemed to move as a homogeneous boundary or slightly heterogeneous boundary was sort of the extent of the information that you got. Now, starting in the 50s and early 60s, the Swedish labs and a Swedish company started making polymers that we could use for gels. So the earliest materials that we did electrophoresis on were literally paper. Okay, and I think I have these slides. I have two slides from different versions of this. So let's go to the more complicated one. And literally a piece of paper, a big piece of paper. It usually was fairly wide and fairly long. And I'm talking like feet, 4 or 5 feet long. It would be wrapped between electrodes. So there'd be an upper sample and a lower sample where the buffers were that established the field. And then you would put spot stuff on the paper and it would migrate. Okay. And typically this was done with things like amino acids. So if you wanted to do amino acid composition, you would hydrolyze a protein to its constituent amino acids, and then you would dot them or blot them on the paper, and then they would run, and you would get spots at different positions that had something to do with the mobility of the amino acids. And then you would stain it with something that stained amino acids, ninhydrin or something. And you could, upon calibration and running standards, learn something about the composition of a protein. Okay. There are clearly problems in this technique that had to do with evaporation because it was exposed to the atmosphere. And then endoosmosis basically has to do with weird sort of electric currents that would get set up in the sheet because of inhomogeneities in the paper. The next system that was used was something involving starch. I don't use starch for anything. I guess some people use starch for cooking or things like this, but it's basically a solid, I've actually poured starch gels many, many years ago. They create sort of a solid matrix. You have to create a little well in them so that you can add a sample. And then you, again, have some kind of a electrode, and it allows samples to migrate into the starch. There's a figure in the text that I think we're going to see in a few moments. It worked pretty well, but basically the starch created a porous gel, and the size of the pores was sort of one size fits all. All you got was whatever the starch gave you. And so it didn't have a lot of resolving power. This says high resolving power, but it really didn't have the ability to fractionate by the sieving effect that we're going to see in polyacrylamide gels. Recovery, removal of samples difficult. Recovery of samples was difficult. Agarose gels or agar gels then also came online, although much after, I think, starch, and they tend to give more open pores, which made it similar to free electrophoresis that I described up above. They work best with DNA, and that's how most people run their DNA samples, in part because the pores are so big and it allows large molecules to migrate. Notice this is saying here that the gel in agarose tends to be negatively charged. So that may be one of the reasons that it works better with DNA than with protein. Okay. But eventually in the 60s, somebody created something called polyacrylamide. And polyacrylamide is a polymer that will polymerize upon activation with the right catalysts and in the presence of the right cross-linkers. There's something called bisacrylamide that does the cross-linking. And you can make the acrylamide in different concentrations, so you get different pore sizes. Okay, and so you could get much higher resolution and much better fractionation. And it wasn't until the mid to late 60s that acrylamide gels became a popular thing to do. And I'll say a little bit more about that in a few moments. Okay, so this is a general example taken from the book, chapter 5, a typical apparatus. This is something that is actually sort of late in the process. And what I mean by that is this is a vertical gel. And the early gels were run vertically, but they were actually run in tubes. And I'll show you examples of that shortly. So the well in a typical gel was a single tube where you would cast a gel and then you would load on the top. In a gel like this, it's basically a slab that's run vertically. These, you then, as you pour it, you create some kind of indentation with a plastic mold of some kind that creates the wells, and then you can run multiple samples across it. Again, notice cathodes and anodes and who's running in what direction. All of this depends upon the setup. If the anode is at the bottom like this and you load at the top, then it's the negatively charged stuff. For example, an SDS gel where the negatively charged stuff would migrate into the sample. There's often a dye that also migrates, and it's a colored dye, so it shows you where the front is, so you know when to stop running so things don't run off the gel. There's an upper and then there's a lower gel. Well, excuse me, and these buffers are generally the same, not always. In some gel systems, you actually recirculate stuff because it becomes depleted. Generally speaking, in the polyacrylamide gels that we would typically run today, you don't have to do something like that. Okay, so this is a schematic of a starch gel. I probably should have had this above the other one. That's 6.6, this is 5.2. So I'm mixing media here. So here's an example of a typical starch gel, or what you might call an agarose gel as well. They're generally run flat. So you can see it here as a gel. There's generally some kind of a notch in it where you can load a sample. In this case, it's a continuous notch. Generally, when you pour it, there's some kind of, for the DNA people who do this, there's some kind of a molded indentation that creates little notches so that it has notches across it. I'm going to try to write on here, if it'll let me. So you might have little notches in here so you can load different lanes and you can see different things. So it might be a flat piece of something, there it is, and then it would have these little indentations in it, and it would allow you to create that when you first pour the gel. So it would lay across the top and create these indentations as the agarose sets up, or as the starch sets up. And then you turn on the electrodes, and again, notice we have different charges, so it depends on what we're talking about. If this is some kind of It doesn't actually identify here. Some kind of a electrophoretic mobility that would be attracted to negative charges, then it would migrate into the gel. And in this case, you're seeing two kind of diffuse bands. For DNA, obviously, it's usually something much more specific than that. Let me end this for the moment because I want to show you something. This is an example of something I found on the web. I'm not going to go into great detail with this, but in these gel systems, in the vertical gel systems, for example, and I'm going to come back to this already because I can tell I'm getting ahead of myself. But generally speaking, you can see that there's a buffer in the top and in the bottom. In this case, it's a tris-glycine buffer that I'll tell you about later. But then the gel itself, what is called the separation gel, is poured with a different buffer composition. It's not tris-glycine, it's tris, pH 8.8. So the pH is different. And then there's a stacking gel where you create the lanes. And this also has a different composition and a different pH. And this variation in composition and this variation in pH becomes important later on. Okay, so this is something that you don't do in agarose gels. You only tend to do this in polyacrylamide gels. Okay, so let's go back to this. Now, I'm not going to say a lot about this particular technique, but there's a technique out there that are called Ferguson plots. And these Ferguson plots are basically log of mobility plotted versus the composition of the gel. So this C is literally the concentration of acrylamide. It's actually the total concentration of the acrylamide and the bisacrylamide. And so as you change the concentration of the acrylamide, you change the matrix size. Okay? And so by changing the matrix size, what you're doing is you're causing a sieving action. All right? And so what does that mean? Well, for example, if you had a really small protein, it wouldn't care about the size of the sieves, and so it would not change its mobility upon changing the composition of the acrylamide. So its mobility plot would be flat. All right. If you had a modestly sized protein that sensed the sieving at higher concentrations, then it would have a shallow slope. And as you increase the molecular weight, the slope would become steeper because it is being sieved. At low concentrations of acrylamide, it's essentially free electrophoresis here, free boundary electrophoresis. It has a certain mobility that depends upon the net charge, which of course depends upon the pH. So that's a hidden variable here. But then as it begins to fractionate due to sieving, its mobility will slow down simply because it's impeded by the sieving. And so way over here on the right, it's showing this in a more pictorial way, low molecular weight, high molecular weight. You might all have the same net mobility because you have the same effective charge per unit field, okay? But they would be differentially sensitive to the size. And then this example in the middle is showing you proteins that have exactly the same sieving, so they have exactly the same molecular weight, but they have different mobilities, meaning they have different charge. And in this case, it's suggesting that they're isozymes. They're a protein of a known size, approximately the same size. There's a slight change in amino acid composition, which changes the charge. And so therefore they have different mobilities, okay? And so Ferguson plots are a method. It's not completely out of vogue. It's something that almost no one does anymore. I could talk about it at length because I used to run Ferguson plots a lot in the 80s and 90s. But in any event, you can learn something about molecular weight, net mobility, and charge. And notice this is in free buffer. So this is not with SDS. Now, the kind of patterns that you get are things that I presume all of you are familiar with or relatively familiar with. There's an origin and a bottom to the gel. Excuse me. Notice the positive charges at the bottom. So this is a DNA gel. And then there are multiple bands that you see. And so there's mobility. There's sharp bands here. So notice this is not a diffuse sort of band that it was showing on a starch gel. This is undoubtedly some kind of an agarose gel, but it gives sharp bands having to do partially with the fact that these are restriction endonuclease cleavages, but it's a plasmid. So these are big. They're relatively big, and they're therefore going to run very specific cleavages, but they're going to run with little diffusion simply because they're so big. Okay. The thing that you notice when you do stuff like this is that the mobility or the distance moved from the origin is dependent upon the molecular weight. Okay, and it's specifically dependent upon the log of the molecular weight. So in the text, it says log molecular weight. Down here, it says log number of base pairs, okay, which is proportional to the molecular weight. Okay, and what you see is that the distance moved is linear with the log of the molecular weight. So this was a phenomena that was noticed soon after these kinds of studies were being done. So generally speaking, DNA always has so much charge per base pair. I gave you an assignment about virial coefficients and how the charge was suppressed. But generally speaking, every cleavage, if you do an endonuclease cleavage or a restriction fragment cleavage, every fragment has the same net charge per base pair. And so essentially the net charge is proportional to the size, and that dictates something about the net mobility. Remember, mobility is charge per unit field, okay, or velocity per unit field, excuse me. And so it gives you some measure of what the net charge is on each size. And you can get this linear relationship if you plot the distance moved versus the number of base pairs, okay? So in principle, this is a calibration, and you could learn something about the size of the fragment, all right? So that's the way you should think about it. This is sort of an extra thing that we're gonna talk about because I'm not gonna go into a lot of detail about this. One of the additional things that happens with DNA is that the DNA is so large that different portions of the chain get trapped. And so basically one portion of the chain starts moving through one part of the sieve, and another part of the chain starts moving in another part of the sieve, and they sort of get wrapped around. They get what's called entangled in the language down here. And so they have to disentangle themselves to move. And so the way that this is done is we created something called pulse field electrophoresis, where you pulse the field in different directions. And so notice that they have this going down and up, down and up, down and up. And as you're doing this, it allows the DNA to reorient itself. Okay, I think there's often a little bit of a left-right that goes on as well to help it move. You can set this up for different times and different field strengths. This technique is really used in genomic DNA, literally really large DNA particles. Mostly when you're working with restriction fragments, this kind of thing doesn't happen. Okay. The other thing that happens is that DNA can supercoil. And so the supercoiling also creates different sort of topological problems. And so for DNA, at least, that creates different extensive mobility. Positive or negative supercoiling will potentially have a similar effect, but you will get changes in mobility because of the supercoiling. And so in the presence of things like topoisomerase, you will get a different pattern depending upon how you're retwisting the DNA. Okay, again, this is something that is DNA specific, not protein specific. I think one of the final things, one of the final topics that is discussed in chapter 5 has to do with the fact that electrophoresis is sensitive to shape. Specifically, it's sensitive to curved DNA. So if you have a small piece of DNA, and I'm not giving size here, but if you have a small piece of DNA where there's some kind of a natural curve, and then another piece of DNA that's essentially linear with a smaller bit of curve or no curvature, but of the same essential size, they will have different mobilities because the shape of the piece with the curve will simply have a different shape and it will migrate differently in a gel. And so you can actually see this in gel electrophoresis. When this was discovered, people were surprised by the fact that different base compositions will give different extent of curvature. We're not going to get into what this might be good for. It's interesting biochemistry or molecular biology, but it certainly might have some interesting regulation that affects how different proteins do or don't interact. And so this is an example. A lot of this was done by Don Carruthers at Yale in the early 80s. And they discovered that for different kinds of DNA, so this was a specific sequence, as they cleave the DNA in certain ways across the sequence, and notice this is only going out to 400 base pairs. So this sample, these samples would not have reptation, but they do have this sort of different mobility depending upon where the curving is. You would get dramatically different mobility, and this is relative mobility on a gel. So that relative means relative to the die front. And the mobility that you get would change dramatically depending upon where you cleave the DNA and where the bending was. Okay, and so it was an interesting way of studying this phenomena at the time. Maybe the general take-home lesson is that shape matters in electrophoresis. This is showing a more explicit example. I'm sorry, this should have been moved up above too for the reptation, where supercoiling changes the mobility, and in a two-dimensional gel, and this is really two-dimensional electrophoresis where you have different kinds of drugs that are being intercalated. So these are drugs that bind to base pairs and change their effective mobility, but it's also resolving different extents of supercoiling. And so you can see that you get a different mobility pattern with intercalators and then without intercalators. And you can see this resolution of supercoiling and different sorts of shapes that resolve in different kinds of patterns. So there's a lot of interesting topology that goes on in DNA. Now that example up above that was showing mobility versus log molecular weight, this is a log scale, applies to both DNA and to protein. Okay, and So this is two examples of this relative mobility changing with log molecular weight. And I guess the DNA is open squares and the proteins are closed squares. And so this phenomena applies to both examples. Okay, and so today this is sort of a textbook example. And the example in this particular case, it's not referencing a paper, is talking about histones. Okay, and so Ken Van Hold was a histone guy for a long time. And so a lot of his examples in his books are histones, simply showing you the different size structures that you see when you isolate chromatin. Okay, and so this is an example of that in one of his cases. I had this. I found that other thing on the web and I completely forgot that I already had this example of tris glycine. So let me come back to that slide and let me do something else. Let's take an aside here and talk about this JBC paper. In the early 60s, there was already an observation about this mobility problem, not a mobility problem, this mobility information, that the mobility was molecular weight dependent. And the molecular weight dependence shows up in SDS polyacrylamide gels. So we're not looking at the net, the endogenous charge on the protein. We're looking at the charge on a SDS bound protein. So you use high concentrations of SDS. You usually make up your sample in something like 5% SDS, so you get lots of SDS bound. You usually do it under denaturing conditions completely, so you often boil that sample in the presence of beta-mercaptoethanol, so you don't have any disulfide crosslinks. And the idea is that what you end up with is essentially, come on, there we go. Essentially some kind of a rod that has SDS bound all over the surface. Okay, and I'm not going to try to do this decoration, but it's SDS everywhere that's giving the net charge to the protein. And then internally what you have is a linear a linear protein sequence. And so what you see is the SDS. The protein may have charges along it, but generally speaking, you don't see that because it's crowded by, or surrounded by a net charge of sodium didecyl sulfate. Okay, and the internal binding is done from the uncharged, the decyl sulfate, the dodecyl, carbon moieties, and then the charge is due to the sulfates that are on the surface. All right, and so that was sort of an observation that people had, that it seemed to be proportional to log molecular weight. And so what Weber and Osborne did was they decided to look at this a lot more carefully. And what they did was, it doesn't say, this is the abstract of the summary. There it is. There were 40 proteins that they looked at. This is 1969. And so you need to maybe appreciate that in 1969, there weren't actually that many proteins readily available. You sort of had to get them from other investigators. You couldn't basically go out and necessarily just buy stuff. Sigma was not the company that it is today, so you couldn't do this kind of stuff. So there was sort of a limit on the number of things that were available. Notice it's referring to the original observation by the Mizell group about this phenomena a few years before. And on the right is sort of the basic observation. If you take a mixture of proteins, in this case, it's catalase, Fernerase, I don't even know what Fernerase is. Aldolase, glyceraldehyde phosphate dehydrogenase, carbonic anhydrase, and myoglobin, varying in molecular weight from 60,000 to 17,000, and run them out on a polyacrylamide gel in the presence of SDS. They will resolve as 6 separate bands. So this is obviously myoglobin at the bottom. This is catalase at the top. Okay. And if you rerun them minus one of the components without the myoglobin, then there's no band there. There are some other sort of artificial things that might happen on a gel, but basically you see the same banding pattern. Although notice when you run a tube like this, there's an issue with lining stuff up and deciding where the top is and where the bottom is. Notice how different in length these guys are. So that's one of the problems with tube gels. Okay. This particular gel is not a discontinuous gel. It's a continuous gel. What I mean by that is the buffer composition at the top and at the bottom and in the gel is the same. So that's one of the reasons that the bands don't look sharp. They look sort of fat. Okay, is that there's no impact from the discontinuous gel. I'll say more about that in a minute. Okay. And so this is sort of just the observation that you run 2 gels with essentially the same composition or a slightly different composition. I think they changed something between the gel on the right and the gel on the left. Catalase, I think they leave something out. Liver alcohol dehydrogenase is not in the first gel, but it's in this gel. So there's a slightly different position here. I couldn't actually figure out which was which. So this is a list of all of the proteins that they ran. So this is supposedly 40 proteins, okay? Molecular weights going from myosin at 220,000 to chymotrypsin at 11,000 or 11 and 13,000 because there's 2 chains in chymotrypsin. Okay. So the first thing for me to point out to you is, where do the molecular weights come from? The references are the answer to that. They're measured, but they're not from amino acid composition with exception. I think there are a couple of examples here that might have amino acid composition, but they're not done from DNA sequencing because DNA sequencing didn't exist in 1969. Okay, so we don't really know the amino acid sequence of all of these proteins at this time. They were all measured by other methods. They were all measured by light scattering, or they were measured by osmotic pressure techniques that we've already talked about, or they were measured by sedimentation equilibrium that we'll talk about starting next week. Okay, so these are experimental numbers. And so we know these sizes approximately. Notice that they're not rounded off to single digits. The nearest hundredth seems to be the way they're being rounded off. Okay, so that's the first thing to notice here. Okay, we had to know something about these proteins from other experimental methods. And if you look at the paper more carefully, it'll tell you that. Okay, and then here's an example of the plot where you plot mobility versus log molecular weight. Okay, notice the molecular weight is running from the 16,000 for myoglobin out here for glutamic dehydrogenase, which must be at approximately 54,000. Glutamic dehydrogenase, 53,000. Okay, and it's linear. And they immediately in this figure point out what you can use this for. They had a protein, D-amino acid oxidase, that they didn't know the molecular weight of. And so they ran it on this gel along with all of these other known proteins. and it has a mobility right here. And so if you look on the gel, okay, the dot isn't shown. It's just showing you where the mobility is. It seems to run around 37,000, they say. And they're showing the variation in the mobility, 0.398 and 0.402, and both of these give approximately a value of 37,000. So that's one of the advantages, okay? Now, this is done under a certain set of conditions. There's a certain amount of acrylamide, but there's also a certain amount of crosslinker. And the crosslinker is what really determines the size of the pores. And so on the right is a similar gel with different amounts of crosslinker. Okay, so the middle one is a normal amount, okay, whatever that means in their recipe. The one on the right is 0.5%, and the one on the left is 2%. Okay, so the one on the right is more open, and the one on the left is more closed. And because of the tighter sieving, this more cross-linking and smaller holes basically in the acrylamide, you get more fractionation. And so you begin to lose linearity. All right. You could still use this as a calibration curve, and they discuss that in the paper, but it's curved, it's not linear. So it makes it a little more problematic. All right. The other thing to notice about this is one, whatever normal is, and this is a half. And you get different mobilities, you get different curves. So the issue is when you make up a stock of acrylamide, you generally make acrylamide up with some amount of acrylamide. The way my lab used to make it up, I still have some in a cold box. We haven't run a gel in a long time in my lab. We usually would make a 30% stock. But in that stock, we would also have some amount of bisacrylamide. Okay, so it might be a 1%, and I'm making this up, a bisacrylamide concentration. And then you would dilute this to some amount. So we typically ran 8% gels, although the stack would be a different thing. And so you would do a dilution from this 30% to 8%, and necessarily you would dilute the 1%, okay? So that plot up above where I plotted versus C, that C is the total concentration of acrylamide and bisacrylamide, okay? When you make up a new stock, you also make it up 30%, 1%, but it's not perfect. It's 30.1% and it's 1.05%. And so the ratio is slightly different simply because the weighing is slightly imperfect, and the amount of buffer that you add to it, we usually did this in buffer, you can do it in water, is slightly different. Instead of being 100 mils, it would be 100.5 mils. And so there would be small changes. And consequently, the curves would move slightly. And so if you were making up calibration curves and in the middle, you changed your stock, you would have to repeat everything because suddenly the mobilities are different enough within experimental error that you wouldn't get the same answer. Okay. So here is the summary slide basically for 40 proteins. The molecular weight all the way out, in this case, to 68,000. I think that is probably serum albumin. Okay, it doesn't look like it's the phosphorylase, which runs it at a higher molecular weight. Okay, all the way down again to something around chymotrypsin, 11, 13,000. You can see how linear it is. You can see what the variation is. This error around the line reflects the absolute error in the measurement. We think we know what that molecular weight is, but it's not on the line, it's off the line. So there's some uncertainty. And they talk in the paper about how some of the reproducibility is plus and minus 1 to 2%. Okay, but in general, the uncertainty across all proteins, including unknowns, may be as large as 10% uncertainty. All right. And some of that comes from the fact that proteins that are in an SDS mice cell often are glycosylated. And so it impacts the amount of SDS that can be bound, gram per gram, SDS per protein, and it affects the mobility, contributing to uncertainty. Sometimes proteins are highly charged. They might have lots of phosphate, which might impede SDS binding, which would impede mobility. And so there are lots of reasons why this might happen. Okay. But in general, this technique was about plus and minus 10%. On the right, they're showing you that above a certain molecular weight, this curve necessarily is not linear. They don't have a method that gives you completely linear from 13,000 to 200,000. So you end up having to run a different kind of a gel, slightly different pore sizes, slightly different gel concentrations, and you end up with a curved plot, in this case going from 40,000 to 200,000. You can use this as a calibration. You can run unknowns on here. To extend this to higher molecular weights requires standards. And certainly at this time, there was nothing available above or around 200 to 300,000. Okay, and so this part of the curve is not easily calibrated. So you can do things in this molecular weight range, but there are limitations in the overall molecular weight. Okay. So let me stop for a moment and ask, we're going to take a break, but are there any questions at this point? No questions. Sarah, is that, or Lindsay? Yes.
Okay, my very minor question. On the slide that showed the very first pictorial slide that you showed of the Swedish experiment with the U-tube.
Yeah.
When they showed the three peaks, you know, the three peaks that descended, the three peaks that ascended, or the interfaces, they were numbered 1, 2, 3. Were they insinuating that peak number 1 that ascended is the same thing as peak number 1 that descended on the other side?
Yeah, that was a weird thing about free boundary electrophoresis. You would actually see this kind of weird symmetry of things going in both directions at the same time. Yes.
Okay.
That's sort of a whole topic. And it had something to do with the way that they isolated samples and the heterogeneity in the samples. But yes, you would actually see that.
Okay, thank you.
Yeah. Maybe I'll try to put together some kind of a better explanation of that. In the Ferguson plot, the extrapolated line to 0C was also meant to measure free boundary, because there's no sieving at 0C, 0% C. And so there's some discussion of free boundary electrophoresis when you discuss Ferguson data. The absolute mobility you get there is meant to be thought of as, it's usually called U0. It's thought of as the free boundary electrophoresis. You don't tend to see this sort of weird mixture though, because of the setup of the design. I'll try to put something together that explains that a little better. It's usually shown historically. A lot of people don't show that figure in that experimental design. Ken Van Holt liked to show that. just to give you a rendition or a window into where a lot of this stuff came from. And again, appreciate, by this time, we sort of knew that proteins had a primary sequence, but we didn't really know a lot about heterogeneity and different kinds of compositions. And those samples were typically blood proteins and mixtures of proteins from blood. The IgG fractions would definitely be remarkably heterogeneous. The serum albumin, it was probably pooled blood a lot of times, maybe not always. And so you did have different serum albumins from different people. You would certainly have different hemoglobins if you were running hemoglobins. So there would be some small heterogeneity in the charge. Anything else? So let's take another short break here and we'll get back to it. So just another couple of minutes now. Okay, are we back?
Yes, sir.
I have to share my screen again. Cindy and Matthew, you there?
Yeah, I'm here.
Okay. So I'm guessing that not all of you have run gels. I'm guessing that some of you have run gels. I found this morning. I could have just gone over to the wall in my lab and photographed a recipe for the acrylamide bisacrylamide mixture. Notice this particular recipe is 40% plus 3.3% bis. There's a certain amount of stock SDS that you add. There's a certain amount of buffer that you add. Ammonium persulfate, which is used in the catalyst. The thing that initiates the reaction is this thing called TMED. It's inhibited by oxygen. So you degas the solution, you add the TMED, you degas for another couple of minutes, and then you pour the gel, and then the whole thing sets up. That's the separation gel. These are the different concentrations of the separation gel. And then this is the stacking gel. You'll see here it's 4%. That number varies depending upon how well it's polymerizing and how well it makes the wells, because it's the wells that matter. You want your wells. Come on. It's not going to let me write on here. I can't write on there. That's a web page. I'm going to have to do it over here. You want your wells to be well formed. And this is not drawn really well, but this piece right here that sticks up, this is where the sample goes. But this piece has to be well defined. If it's not well defined, if you get one that doesn't form, then this and this will mix. And so the percentage that you pour is often, for the stacking gel, is often a big deal. Okay, and so you have to worry about that. All right, now I also gave you this paper to look at. This is a description of one of the first, I think the first description of discontinuous gels. Okay, and it describes mixtures of a stock solution. So notice the stock solution is 30%. I told you that's what I use. And 0.8% bis. I put down 1%, but I think this may actually be the mixture. And then notice it's pH 8.8 tris with some SDS. And then he uses TMED and he uses ammonium persulfate. Okay. And then notice that the stacking solution is pH 6.8 tris. Okay, and then the running buffer is tris glycine with SDS. Okay, and so we basically use the Lemley method in my lab. Lots of people use the Lemley method. And I think my lab actually uses the exact concentrations, the exact pHs that Lemley described in this 1970 paper. It also describes the use of a dye, bromephenyl blue, that runs with the front. And then it also describes the sample buffer. Okay, so the sample buffer had some amount of tris. So this is the separation gel was 8.8. The stacking gel is 6.8. So the sample goes in the stacking gel buffer. But then it has an additional amount of SDS, 2% SDS in the sample. And then it has 10% glycerol, and it has 5% 2-mercaptoethanol. So the beta-mercaptoethanol is to break disulfide crosslinks. The glycerol is to give it density so that when you add it to the well, it sinks. If you don't add glycerol, it doesn't sink. And you want it to sink so it sits at the bottom of the well. So let me just show you a well. Okay, so you pipette your sample into the well, and then it literally sinks to the bottom, and you can sort of see it as a Schlieren at the bottom. So it's already sort of concentrated at the bottom of the well. If you don't do that, however much volume you add, it doesn't necessarily displace. You would have to sort of stick that to the very bottom, and it would displace the solution, but it wouldn't settle at the very bottom. It would be diffuse. Okay, and so it already is sort of diluting and spreading the boundary. Now, this is the figure that I had up above. I moved it down below. This is the figure I had up above. And here is the typical Lemley sort of design. There's a pH 8.3 running buffer. There's a 6.8 or 6.7 stack. This figure uses 7. I use 6.8. There's a running buffer of 8.9. I use 8.8, but a lot of people vary the pH because in different buffer situations, you actually get different, slightly different separation. Okay. So the secret of a discontinuous gel is that as it runs, there's a sample at the top of the well, and this is represented, I think here, it says by the sample in the same buffer. And then it migrates through the stacking buffer. And then it hits the higher percentage gel. So this stacking gel might be at 4%, and this gel might be at 8%. So there's suddenly a sieving or a retardation of the sample. And the boundary at the front slows down, and the back edge of the boundary keeps moving until it also hits the zone. So that's a slight concentration that sharpens the bands. But the other thing that happens is that the mobility of the glycine changes in going from 6.7 to 8.9 in this particular case. And suddenly the glycine zips ahead and it moves faster in the lower gel than in the stacking gel. And that increase in mobility at that interface pulls the protein along and sharpens the band. All of the back part of the zone is pulled along in this flow of the higher mobility, and it sharpens the bands. And so the big advantage of discontinuous gels is sharp bands. Okay, it's achieved by the stack separation a little bit. It's mostly achieved by the fact that the mobility of some of these ions changes from low pH to high pH, and that causes the sharpening of the band. When the band first enters the gel, you can often see it, physically see it, because it's so sharp and so concentrated, it diffracts light. And so light coming in will bend off of it, and you can actually see this sharp band, especially for the high molecular weight stuff. And then it will diffuse as it runs deeper into the gel. Okay, and so that's what Lemley did. Okay, now I don't think I have any figures. Yeah, I don't have any figures out of Lemley's paper. The application of this was a Nature paper, I believe, right? And so the application of this is looking at structural proteins being expressed and modified during the assembly of a bacteriophage, T4. And so it's using different it's using different sort of protocols to watch the development of the virus as it grows. And it's the resolving power that gives the power of this technique. So let me actually go get that paper so I can show you at least one gel. Okay, and so it's Lemley, Nature. And so notice the sharpness of the bands. Okay, if there's a high concentration, it will be broader, but notice that the lower loads are reasonably sharp. They smile a little bit. That smiling has to do with the fact probably that they're running these gels a little bit too fast with too high a current. And so there's a little bit of a heating artifact that occurs. And so the middle of the band runs faster than the edges of the band. Okay, I think these may actually be tubes as well. So I think these are lined up. These are not just lanes on a discontinuous gel. And so that's a little bit of what's going on here. But you can actually see different sorts of bands that don't occur, bands that occur. You can actually watch things evolve with time. So notice, I think this is time up here, when they induce things and when different things form, et cetera. Okay, and so that was the sort of the application and the power of this. But the utility of it was really enhanced by the gel system and the discontinuous gel system. All right, and if you had just run a continuous gel like in the Weber and Osborne paper, you wouldn't have had the resolution that was achieved with this. There are lots of discontinuous gel systems out there. You can imagine all kinds of different combinations of pHs and buffer compositions. Okay. The Lemley technique is the one that is, I think, most broadly used. And as I say, my lab continued to use the Lemley system I use it as a postdoc. So I've used the Lemley system for 40 years. Okay. There's a section in here that discusses blotting and the fact that you can blot fragments of DNA onto some kind of this is, I think, the gel with the fragments blotted onto some kind of nitrocellulose filter, and then visualizing in some way, shape, or form. You can then hybridize to see if the fragment corresponds to a particular sequence. This looks like it's taken out of one of the early versions of Watson's book, Molecular Biology of the Gene. This is the book you guys use in molecular biology or in cell biology, I guess, except it now has a half a dozen authors because Watson hasn't been an author of this book for decades. And it's just showing you the different kinds of things that you can do for blotting. And it mentions the fact that a southern is for DNA, a northern is for RNA, and a western is for proteins. And using these various techniques for identifying particular fragments. There's some discussion in chapter 5 in the Van Hol Johnson Ho book of the utility of this. The history chapter tells you where the names basically came from. Not everybody uses gels. You can use other kinds of apparatuses that will do electrophoresis and so show fractionation of proteins along capillary electrophoresis. So there are other sorts of matrices that you can do for similar kinds of things. This is set up with injectors. This is probably monitoring at some wavelength, so it picks up the protein. And so that's another way that lots of people do electrophoresis. I've actually never done electrophoresis in a capillary apparatus, but I think I mentioned it in the medical biochemistry class when we talk about hemoglobin and the various mutant forms of sickle cell that we talk about, that some people use other kinds of fractionation techniques. I think one of the final techniques that this chapter talks about is something called isoelectric focusing. Okay, and so this figure is sort of dictating what's going on. You have some kind of a gel. We used to pour gels to do this in my lab. We would have really long gels, 20, 25 centimeters long. And then you would add to the gel mixture, and then in the upper and lower reservoir, you would add a solution of something like 1 to 2% of compounds that have different isoelectric points. So these are called amphylines, also primarily made by these companies in Sweden. And the amphylines would establish a pH gradient because they would go to a pH where they don't have mobility. Okay. And then you would add a protein. In my case, tubes at the top, you would add a sample. Depending on how you're running it, you don't necessarily have to run it at the high pH or low pH end. Many samples would denature if you made them cross their isoelectric point, so you had to be careful with loading. And then it would migrate to some isoelectric point where it had no mobility. And so it would tend to stop at those places. Okay, and so this is sort of depicting how different molecules across the gel would migrate in different dimensions, and over time it would eventually line up at its isoelectric point with some kind of width to the peak. We have a flat apparatus, which is basically a strip that has a gel poured on it, and the gel actually already has a pH gradient on the gel. It's actually cross-linked to the gel. You still add amphylines to it to reestablish it, but you actually pour your sample along the entire length of the gel, and then it migrates to its isoelectric point. So this is a depiction in Van Holt of what that might look like. This is hemoglobin by isoelectric focusing. Here is the pH gradient. In this particular case, they had to break the gel up, and the slices were put in the water, and then the pH was measured to establish what the gradient was. And then the protein gave various peaks. These might be scanned optically. These might be a different kind of depiction. I think this is scanned optically in some kind of a reader in a spectrophotometer. We would stain them and photograph them. I'll show you an example in a second. But you can see the various peaks that you see and what kind of resolution you're getting. And notice this resolution is from 723 to 764. So there's a lot of spreading out or smearing of these 1, 2, 3, 4, 5, 6 bands. Okay, presumably different forms of modified hemoglobin or mixtures of different kinds of hemoglobin. This figure is meant to show that the earlier derivation that we did on the mobility and the use of Henry's function and the Bihuckle doesn't perfectly match the shape of what you actually get in mobility. But over narrow regions, it's an accurate description of what you get. There's a lot of deviation at the edges. There's a lot of bleeding off of the edges of these gels, and you run them for a long time, overnight very often. And over time, the gradient sort of just diffuses and electrophoresis away. So that's partially what this is showing. Here's an example that Ken stole from a book by a guy by the name of Finn Vold, showing you examples of isoelectric patterns and how sharp the bands might be and how reproducible the bands might be. Now, that's a one-dimensional gel, okay? And so sometime early in the history of all of this, 1975, O'Farrell decided that, you know, if I ran an isoelectric focusing gel and then I turned it on its side and I ran an SDS gel, I could identify what these bands are by their molecular weights. So I could run a charged gel, isoelectric point, and another dimension for a molecular weight, and I could identify bands this way. So 2D isoelectric SDS gel methods then became the vogue. All right. Now, O'Farrell's paper was also in JBC in 75. And the summary is shown here. You can go read the paper and look at that. This is a depiction of what the gel apparatus sort of looked like. You ran a tube, and the tube then laid down in a notch. I don't know if it's obvious to you that that's a notch up there, but there's a notch that the gel sort of lays down on so that the length of the tube is across the top. When you run an isoelectric focusing gel, you tend to run it in really high concentrations of a denaturant like urea. And then you have all of these other things that are in there. And then most importantly, the proteins are at their isoelectric points. So one of the things you have to worry about is precipitation, because proteins will precipitate at their isoelectric points. So what you have to do is take this gel and sort of equilibrate it in an SDS stacking buffer for some period of time to make sure that the proteins are soluble. And then you lay it on the second dimension. You have to know what the bottom and the top is, so you know what the directions are. You don't want to flip things around so it's upside down. You sort of want to know where you are in the pH gradient. Normally what people do is they pour some kind of a stack around it and keep it in place, so it's sitting flat on the bottom. Usually you pour some kind of a low concentration agarose solution so it's solid and things don't sort of drift out of place. And then you run the SDS dimension. So this is an example of an isoelectric focusing gel run in a second dimension with SDS. And this is an extract of something like E. coli. And then this is something that's radioactive. So these are actually transferred to film. So you probably lay a piece of film on the gel and you allow it to irradiate the film for some period of time. I think these irradiations were done over the course of a month. And you can see how many spots that you see coming out of the total E. coli genome. All right. So you can see maybe, I think he says in the paper, up to maybe 1,500 different spots. In principle, you should be able to see something like 10,000 spots, but that depends upon how many proteins you have with different isoelectric points and different molecular weights. Okay, and so this begins to show you something about the resolving power of this method. You can see lots of different stuff here. Now, identifying what it is begins to become part of the problem. But the question is what are each and every one of these spots, and do they change under certain conditions? Allows you to do interesting kinds of science. So this is a methods lecture. And so that's primarily what we're interested in. But what he's showing is that when you run a one-dimensional SDS gel, you can see all of these bands. So you can learn something about them. But then if you move to the right, you start going, okay, but this is their molecular weight, but what's their charge? And you can begin to identify that this region of the gel actually has 3 or 4 or 5 bands that resolve in a focusing dimension that don't necessarily resolve in a molecular weight dimension, okay? You can occasionally have weird sort of smearing artifacts on the edges. You can see that some of these bands look like they're smearing a little bit, and that often is an artifact that you can sort of resolve. You can see some bands where there's sort of an obvious doublet, and this may actually indicate post-translational modifications. It could also indicate an isoenzyme or a different gene product. Same protein, slightly different charge, but same molecular weight. Okay, and so that's something that you can see in a gel like this. Now, the paper itself gets into things like the role of loads, how much protein do you load, and the resolution of the spots and what the shapes of them are and how they impede on one another. There's a lot of different questions that are answered in the paper, I think there's something like 13 figures that go through all of these different kinds of answers. Here's post-translational modifications. You can actually see a mutation that might appear in a particular example. You can see some examples of unusual artifacts. So there's all kinds of things that the paper really talks about. So starting in 1975, lots of people started doing 2D gels. You would run a focusing dimension. The pH range over which you ran it depended a lot on what you were running. These amphiolites that you buy can be broad amphiolites, they can be narrow amphiolites. We did mixtures of them. We were primarily using them for tubulin, which is a fairly narrow band in pH. If you're running whole extracts and you have to do a fairly broad pH range, maybe 3 to 10, okay? And then narrower ranges for different kinds of proteins, 5 to 7 or 4 to 6, something like that. And we usually ran mixtures of things like 5 to 7 because the tubulin isoelectric points were in that region. Okay, and so I'm just going to show you one sort of application because we were interested in cleaving off. This is, you can see here, this is 30 years ago. We were interested in cleaving off a tail that exists in the carboxy end of tubulin that has lots of post-translational modifications. And so we were interested in looking at post-translational modifications. So these are examples of native and subtle isin cleaved polymers. Lane 1 is native. And then when you cleave off the carboxy tail, then you can see a shift in the isoelectric focusing patterns and a change in the banding pattern that you get. Okay, so this is a one-dimensional isoelectric focusing. These are tube gels with a gradient. Okay, there are different samples here. I won't get into what all of this is. Okay, but then we actually started assigning values. Tubulin is a dimer. Okay, and so we started looking at what bands we get from alpha and from beta. Okay, these are different tubulin isotypes that we're looking at. So we're actually trying to isolate the different isotypes of tubulin. Tubulin actually has multiple alpha genes and multiple beta genes, and depending on where you isolate the samples, you will or will not see different kinds of banding patterns. All right. How do I know it's alpha or beta? They're clearly different sequences. Tubulin's an alpha beta dimer, but if you run them in a 2D gel, okay, the alpha and the beta run at different molecular weights. The different molecular weights reflect something about the composition of the alpha beta bands, and they reflect something about the post-translational modifications of those different bands. So these are sort of a little bit smeared, but you can see that there's a number of different isotypes in here. Okay, it's hard to count them in this dimension. It's a little bit easier to count them when you look at them like this, although even these are a little bit smeared. But this one is native. So here's the native region of the gel. Okay. And then when you cleave it with subtle isin, the beta chain is cleaved first usually. So it starts to go away first. The alpha chain is cleaved less quickly. And you can see it resolve into different bands more readily. And so what happens is you cut off the carboxy tail, and now you're down to the base sequence without the last 20 or so amino acids. And then you can identify different bands corresponding to subtle isin cleaved beta or alpha tubulin. Okay, now in this series of papers, we actually could isolate individual isotypes by various techniques involving affinity columns, and then subject them to exactly the same sorts of patterns. Okay, the reason that there's a smear is often because the carboxy tail has various post-translational modifications. They're usually polyglutamylated. So there might be 1, 2, 3, 4, 5, 6, 7 glutamates, which adds charge, obviously. So the smearing is partially isotypes and partially post-translational modifications. It's interesting if I showed you the same sort of pattern, which I'm not going to, a subsequent paper from a single isotype isolated from cells, then we discovered that various cells have fewer isotypes and fewer post-translational modifications than the brain tubulin that we normally worked on. And so that's the power of these types of techniques, okay? Tubulin was unusual because if you really ran a high resolution gel done at its best, there are more than 30 bands here in a pH range of something like 0.3 or 0.4. And so the resolution of 30 bands was really difficult. You had to really put a high concentration on the gel, and you really had to have great gradients to be able to resolve this. We did this back in the day, but there were other people who were better at it. This is what led to the development of stable gradients so that it was preformed, that made the resolution a lot better. That's all sort of early 90s, mid 90s, late 80s, early 90s sort of technology. Okay. So I'm going to stop. I'm going to stop sharing and we're going to come back here. Okay, so more questions. Okay, so if you look around this department, I don't actually know what everybody does in terms of gels. I'm only familiar with my lab, which used to run gels all the time. We don't run gels at all anymore. In the early 90s, right through the 2000s, that's what we ran. You do or don't know that I'm consolidating. I'm putting everything in G208 into G209. So I'm having to throw away a lot of stuff. And one of the things that I'm throwing away a lot of are gel apparatuses. I have multiple generations of gel apparatuses dating back into the 80s that nobody is interested in. So I'm trashing beautiful gel apparatuses, where you pour them, how you set them up and run them, how you blot them and stain them. I think the Brown lab is the primary lab for doing DNA gels. They're primarily looking at restriction fragments as far as I know. So David runs lots and lots and lots of agarose gels. They're usually flatbed apparatuses. The Abear lab does a lot of blotting. So they do a lot of transfers where you take the gel and you put it down on something and you transfer it in some way, shape, or form. Transferring of DNA onto nitrocellulose or something like that is relatively easy to do and quick to do. My lab did a lot of blotting with proteins, and so you had to transfer it onto some kind of a surface. Nitrocellulose works, but then we would use an antibody to identify which band was which. The antibodies, by the way, are specific to the carboxy tails that we were cutting off in that other paper that I showed you. So we have various kinds of blotting techniques. We used to have I still do have the box for vertical. So you would set it up so the gel laid in a vertical apparatus and you would transfer onto the surface in this kind of vertical apparatus. It was primarily a protein technique, but what we found was it really worked better on a flatbed. And so you would literally make these funny sandwiches of the gel and the blotting paper, but then filter papers on top and the bottom to kind of hold it all together. We used to do a lot of that, mostly in the 90s, some in the early 2000s, depending on what we were looking at the time. So I think it's mostly Michael's lab who does a lot of blotting. Almost nobody does radioactivity anymore. Radioactivity was what Farrell used. I think this department has no radioactive labs. I think that's true. I could be wrong. So it's mostly some kind of a secondary antibody that gives you a color. And those are the primary techniques that people are using. But even that is sort of starting to go away. We used to have rooms at sort of that end of the hallway. And so I'm pointing towards, I guess I'm pointing east here. So sort of that big parking garage down near the library. But at this end of the hall that we used to have multiple rooms for photography and for film development. And I think those rooms still exist, but I'm not sure that the machines still exist for film development the way they used to back in the day when lots of labs were doing this in the building. The whole technique is sort of gone out of, not so much out of favor, is just people don't use it in this lab, in this department anymore. So mostly this is a very meant to be a practical lecture. I will make some reference to it when we start looking at sedimentation, because we'll derive an expression for velocity per unit field. So we will derive an expression of the mobility in a sedimentation experiment. We will call it the sedimentation coefficient, but it's mobility or velocity per unit field. In this case, it's the field strength of a rotating rotor. And then equate it to some information. In electrophoresis, it's charge per frictional coefficient. So it moves fast because of the net charge, but it's impeded by the frictional coefficient. In sedimentation, it moves fast because of the molecular weight, but it's impeded by the shape, the frictional coefficient. Okay, and so it's a similar sort of theory, a similar sort of application. And we will see similar sort of patterns. If we took derivatives, we would see peaks. We tend not to take derivatives anymore. We just plot raw data and we see some kind of a migrating boundary. And we study the boundary and the heterogeneity of the boundary and the different kinds of boundaries and the concentration dependence of the boundaries. So any questions at all? All right, so I'll put an assignment out there. I'm looking at the two chapters, deciding what I want to assign. At the moment, I just have some questions out of the chapter 5, the book. I think it's the last few questions because that chapter discusses more than electrophoresis. It's just the last few questions. One of them is, here's a series of standards. Fit it to a straight line, put your unknown on it, and read out the molecular weight. So it's how you would do that sort of mobility versus log molecular weight analysis. You buy the standards as a mixture. You can buy them in a low molecular weight range or a high molecular weight range, and it applies to various types of mixtures. You can even buy standards that have pre-color on them. So you don't have to stain them because they already have color on them. They've already been marked in some way, shape, or form with a different colored dye. In principle, to save you the trouble of staining them. You still have to stain your unknowns. So I'm never sure exactly what the advantage of this is, because your unknowns, you definitely have to stain with something. I didn't talk about that hardly at all. The Lemley paper talks about the use of different kinds of stains. My lab primarily uses Kamasi Brilliant Blue. I've known other dyes in other labs. Kamasi tends to be the main dye that we use for binding to proteins. For DNA, you primarily use Ethidium, but I'm not sure if that's really what everybody uses. I know David uses Ethidium. It adds excitement to your life because Ethidium is carcinogenic. So you don't really want to play around with Ethidium. You should wear gloves when you're working with Ethidium. Okay. You're not supposed to allow the acrylamide to touch your skin either, because acrylamide is a neurotoxin as monomers. So you're always told to wear gloves with acrylamide. I can tell you for a fact that my lab never wore gloves. We were just careful not to touch the monomer solution. There might have been some laziness involved. Gloves are a little bit cumbersome to work with. All right, no questions? All right, so remember, we're talking about a session on Friday. Two people are away. They won't be back until Thursday, I guess, Wednesday or Thursday at experimental biology. We didn't talk about a time. I'm looking at my calendar. I have a I have lots of stuff here, but it's not anything that I'm going to go to. There's a baby shower here. I probably won't go to that baby shower. There's a couple other things here that I'm not involved in. They just show up in my calendar for some reason. Okay, so morning, noon, afternoon. Do we have opinions?
My opinion, Dr. Correa, would be around noon. I've got a pretty interesting talk that I'd like to attend online from 10 to 11, but after that. So, I mean, all other things being equal.
So there are 4 of you who haven't spoken. Do you care? Lindsay is suggesting something around noon.
I'd be good with any time.
That's 2
Yeah.
pluses. Matt
I'm
Schmirk.
good with at noon too.
Ahmed,
If possible, morning.
say again.
Morning.
You're saying morning. So Lindsay has something at 10. So morning would mean 11.
Yeah.
Is 11 better than noon for you, or does anybody else care? Or a little after 11?
11 is good.
Okay, is that okay, Lindsay?
That works fine. That'd be great.
Okay, I mean, we could do it 11:05 or something to make sure you have time.
It's a virtual meeting and there'll be questions. I can dodge out of the end of the questions.
Okay, I'll send out a Zoom, but the Zoom will include a question so that the other 2 people can express objections. All right, and so tentatively, we're going to do 11:05 on Friday. The goal of this is to go over assignments, to go over things that you haven't handed in or you need some more help working on that you've already handed in, but I've graded them and you're having some trouble with them. It's primarily meant to get your skill sets up. A lot of you are behind in this sort of thing. I know a lot of you are waiting for perfection. You're waiting for insight. And so we need to start sort of getting stuff in so that we can make some progress on all of this stuff. And so I think that's why we're going to have to set up a regular thing to go over some of the problem sets. It'll definitely not be more than an hour. We'll try to make it concise as we can. Okay? Anything else? All right. I'll see you Friday at 11. That's the tentative plan. Bye.
Thanks, Dr. Correa.
Thank you.
Thank you.